Dear student, myself Jitendra Kumar Verma, Assistant Professor in Botany in your college. जैसा कि आपको याद है तो पहले भी हमने क्लासेस ले रखी है आपकी कि हम BSc पार्ट सेकंड में ये पेपर पढ़ा रहे हैं। ये वाला आपका सिलेबस है जिसके अंदर हम यूनिट 3 इस टाइम ले रहे हैं, जो है मेजर कंट्रीब्यूशन ऑफ साइटोलॉजी, फाइटोकेमिस्ट्री, टैक्सोमेट्रिक्स एटसेट्रा। तो आज हमारा टॉपिक का टाइटल है मेजर कंट्रीब्यूशन ऑफ अदर ब्रांचेज ऑफ बॉटनी इन प्लांट टैक्सोनॉमी। ये बेसिकली 3-4 टुकड़ों में करके ये पूरा पॉइंट कवर होगा। अभी हम जनरल वे में लेंगे, देन साइटोलॉजी, फाइटोकेमिस्ट्री, टैक्सोमेट्रिक्स, इनका योगदान इसमें देखेंगे। टैक्सोनॉमी इज द यस्टरडे, टुडे एंड टुमारो। Historically, plant taxonomy starts so simple as the need of the people to those plants. Gradually, this branch begins to be in the form of the scientific works, as plant identity, distribution, morphology, and physiology were emphasized. With the invention of the microscope, scientists were able to see small processes, internal structure, development, and the stages in cell division became an important subject for plant study. Historically, paleobotany concerned the evaluation of the progression of vegetation in a given location. As the environment changed, it dealt with the slow changes in vegetation in a given region. The evolution of various taxa through geological time and evolution of life. Thus, the field of plant taxonomy were active. Evolution was approached by assuming the plant were adapted to their environment. With the discovery of basic principles of inheritance and evolution by natural selection, the science of genetics and evolutionary biology flourished. There was much fascination with the development of the common morphological traits in similar environment across phylogenetically separate taxa. Thus, the investigators of the paleobotanist provided a firm foundation for the plant biology. Within the last 50 years, plant taxonomists have begun to look for more precise technique in order to understand the relation between the genera and families. A lot of questions have been brought in mind, which is more advanced and which are less advanced. What is the origin of plants which more related or which must kept apart? And many other questions are kept in mind. These have made the taxonomists to go further inside the internal structure of the plant, and especially the chromosome structure, DNA sequence, genome structure. This was this what we call the molecular taxonomy. Data obtained from this study are compared with those obtained from the tools of other tools of taxonomy, and all these data have used the cladistic analysis and phylogenetic interpretation. In the second half of the 20th century, molecular biology revolutionized the study of plant at all levels by discovering the structure of DNA, RNA, steps of protein synthesis. Taxonomists, evolutionists, cytologists, physiologists, and developmental biologists are now using molecular techniques and are discovering many responses and mechanisms that were not accessible in the past. It is now possible to identify with much precision the particular gene responsible for traits. As well as with molecular techniques, scientists can introduce or eliminate genes for specific trait. Using these advanced techniques, we may also alter the present taxonomy and phylogeny, and the differences and similarities among taxa are modified by human action. We can create new species. Ecology will become more important in taxonomy than before, as the changes in environment through the coming years will lead to the extension of some species and appearance of new taxa. Broad niche species will dominate while narrow niche ones were eliminated. How much of our plant can cooperate with environment disorder and state new more stable taxonomic character? Gene bank for the narrow niched species must be done. We have to gather all the data from the all aspect of taxonomy beside those derived from the molecular studies and ecological changes in order to understand the relationship between taxa. तो टैक्सोनॉमी इन्हीं चीज़ों को यूटिलाइज करती है, इसलिए मॉडर्न टैक्सोनॉमी जो है एक मल्टी डिसिप्लिनरी ब्रांच अप्रोच को फॉलो करते हैं। यही हम इस एरिया में पढ़ेंगे भी। जैसे अल्फा टैक्सोनॉमी इज बेस्ड ऑन द एम्पिरिकल अप्रोच, वाइल द मॉडर्न और ओमेगा टैक्सोनॉमी फॉलो एज इंटरप्रेटिव अप्रोच यूजिंग इवोल्यूशनरी information to understand taxonomic and phylogenetic relationship. This involves the usage of the knowledge from the biosystematics or experimental phase and the encyclopedic or holotaxonomic phase in taxonomy. Morphological and anatomical characters can be studied with light as well as electron microscope, and this information is useful for solving taxonomic problems. Plant embryology has proved extremely useful as source of evidence in taxonomy. Cytology, biosynthetic data use information from chromosome number, structure, behavior. Chemotaxonomy provides data from the chemical characteristics. Taxonomists can use computers to analyze large amount of the information in numerical taxonomy. तो इन सब ब्रांच के एडवांसमेंट की वजह से टैक्सोनॉमी की अप्रोच में भी चेंजेस आए। Today, the modern taxonomists realize that the ultimate taxonomy of higher plants must be based on the understanding of morphological, anatomical, embryological, cytological, and breeding behavior, besides biochemistry and other multidisciplinary approach. Morphology in relation to taxonomy को अगर हम देखें तो morphology is the basic tool of taxonomy because it identifies the primarily based on the character of the plant. The morphological characters are easily observable in both living as well as the herbarium species. They are provide the basic information. Majority of the classification system in plant taxonomy are based on these. In recent years, electron microscopy has provided a valuable tool to the modern taxonomists to study different morphological characters at higher magnification. This information used the purpose of identification, classification, and for establishing relationships. Then the use of the anatomical characters in taxonomy begins with the development of the microscope. It was realized that the anatomical characters are just as valuable as morphological one. We can also know that this subject is of great importance to scientists who are called upon the all samples scraped the plant material for particular purpose, such as the pharmacologist in the determination of the source of drugs, or by forensic expert who may also be able to provide the clues of the crime investigation. फिर इसके अलावा अगर हम देखें एम्ब्रियोलॉजिकल इंफॉर्मेशन की तो इनको भी टैक्सोनॉमिक पर्पस एट द वेरियस लेवल ऑफ क्लासिफिकेशन यूज ले सकते हैं। You know the basic division of plant kingdom into two parts, thallophyte and embryophyte, where the plant are recognized on the basis of zygote. जब हमने शुरू में देखा है कि वह प्लांट का बेसिक डिवीज़न भी एम्ब्रियो पे आधारित है, तो इसके अदर कैरेक्टर्स को भी हम टैक्सोनॉमी में यूज ले रहे हैं। अब इन कैरेक्टर्स में क्या है? एम्ब्रियोलॉजिकल कैरेक्टर्स में है आपके एंथर के, क्वाड्रिपार्टीशन ऑफ द माइक्रोस्कोप मदर सेल है, पोलन ग्रेन्स के कैरेक्टर्स हैं, डेवलपमेंट एंड स्ट्रक्चर ऑफ ओव्यूल है, ओरिजिन है, स्पोरोजिनस टिश्यूज जो ओव्यूल में है कहाँ से ओरिजिन है उसका, मेगा स्पोरोजेनेसिस एंड डेवलपमेंट ऑफ एम्ब्रियो सेक है, फॉर्म एंड ऑर्गेनाइजेशन ऑफ मैच्योर एम्ब्रियो सेक है, फर्टिलाइजेशन है, एंडोस्पर्म में एम्ब्रियो है, सीड कोट है, उसकी लेयर्स है, और स्पेशल फीचर अगर प्रेजेंट है तो उसको भी हम एम्ब्रियोलॉजिकल डाटा की फॉर्म में टैक्सोनॉमी में यूज ले रहे हैं। केमोटैक्सोनॉमी में क्या है? ये साइंस, दिस इज द साइंस, केमोटैक्सोनॉमी इज अ साइंस व्हिच यूजेस द केमिकल इंफॉर्मेशन एज अ कैरेक्टर फॉर द टैक्सोनॉमिक पर्पस। Before we analyze the basic of modern trends in plant taxonomy, let us a moment think about the different kind of plant in our daily life. When we drink tea, coffee, we appreciate the flavor and aroma, differentiated by the two by its character. Similarly, when you eat fruit like mango, banana, apple, we find the taste is differently. The potential importance of the chemical evidence in plant taxonomy has been suggested by both botanists and chemists, and this has become an important in recent years, especially because newer technique for quick analysis of plant material have developed. इसी तरह से न्यूमेरिकल टैक्सोनॉमी टुडे इज ऑफ मैनी डिटेल्स different from that it was a generation ago. This because computers by taxonomists has established the interpretation modern called as the numerical taxonomy as the taximetrics. Mathematical and statistical evaluation of the taxonomic information and computation of the data has provided the taxonomist with new approach to understand the classification. इस तरह से हमने देखा था पहले भी कि ऐसे एंजियोस्पर्म का लाइफ साइकिल है, तो ऐसे अलग अलग फेजेज के कैरेक्टर्स को हम इसमें यूज लेते हैं. जैसे हमने कहा यहाँ सीड वाले कैरेक्टर्स को हम यूज लेते हैं, ये मॉर्फोलॉजी के ले सकते हैं, और इसके अंदर हम एम्ब्रियोलॉजिकल डाटा में अलग अलग पार्ट्स के कैरेक्टर्स को हम यूज लेते हैं टैक्सोनॉमी प्रॉब्लम्स में. जैसे पोलन ग्रेन है उसको भी यूज ले लेंगे, इधर ओव्यूल का ले लेते हैं, इधर मेगा गैमेटोफाइट की पोजीशन है, चलेजा है कि है, माइक्रोपाइल कैसा है, पोलन ट्यूब की एंटी, वो भी एक क्राइटेरिया है. द क्लासिफिकेशन वाज स्टार्टेड विद द कैटेगराइजेशन ऑफ स्पीसीज ऑन द बेसिस ऑफ देयर मॉर्फोलॉजिकल कैरेक्टर्स. The flower morphological characters are majority used by the classification. Besides floral, other morphological characters also play an important role in identification of species of the group of plant. In recent era, the development of science, different branches of botany have developed. These branches are used as the modern tools to give a systematic position and examine phylogeny relationship. The anatomical work keeping taxonomy in view of the mainly carried out by the Bailey Quest was described trends of the xylem evolution, especially for the primitive angiosperm. The chromosomal characters and their behavior during the cell division provides relevant information for the species identification and their systematic position. That chemical composition of plants play a very important role in plant identification. There are the various chemical compounds which are used to establish taxonomy, such as the alkaloids, amino acid, betalains, fatty acid, carotenoids, flavonoid, polysaccharide, tannin, terpenoid, etc. The DNA hybridization, DNA polymorphism, and electrophoresis are important molecular techniques to study at the molecular level. To sort out the systematic problems and phylogenetic relationship among different taxa in Triticum, Hordeum, and Sickle, the results of DNA hybridization show that the Triticum is more closely related to Sickle than the Hordeum. Hawks and Turkers observed a strong serological relationship between Solanum, Nicotiana, Lycopersicum, and Datura. Then morphology. When the classification studied were initial phase, the gross morphological character used for the categorization of species, such as the earlier classification like the artificial natural system, floral morphology, and are majority for the classification. So, and beside their other morphological character also play important in identifying the specific group of the plant, like the anatomy. Anatomy work keeping taxonomy in view are the mainly carried out by the Bailey and the co-workers. Metcalfe and Conquest has described the trends of the xylem evolution, especially for primitive angiosperm. With the advancement of the science, the anatomical characteristics have played an important role in solving the taxonomic problems and understanding the phylogenetic relationship between different taxonomic groups. Then embryological and palynology. The study of structure of the pollen grains is very significant in plant taxonomy because every genera has a specific distinct pollen wall. This has helped to establish the evolutionary study of angiosperm. The taxa show single morphological pollen type is considered stenophilous, while the taxa which have the different of the type of the pollen grains are considered the Euryphalians. The stenophalinous group are of considerable importance in the plant taxonomy. Then इसी तरह से हम साइटोलॉजी के केस में भी देखें तो कुछ एस्पेक्ट जैसे क्रोमोसोम नंबर, मॉर्फोलॉजी ऑफ क्रोमोसोम, बिहेवियर ऑफ द क्रोमोसोम ड्यूरिंग सेल डिवीजन, इनका भी योगदान है. फाइटोकेमिस्ट्री का आप देख ही चुके हैं. इसी तरह से नई टेक्निक है इलेक्ट्रोफोरेसिस. The electrophoresis is an important molecular technique to study mainly the proteins and proteins and others to separate and identify the proteins. The amphoteric properties are based on the to separate the proteins due to the positive or negatively charged in the various extent. According to pH of the medium, protein travel in the gel at the various speed across the voltage gradient. This reaction is carried out by the PEG, polyacrylamide gel electrophoresis. DNA and RNA hybridization को भी आजकल काम में ले रहे हैं. DNA is extracted from the organism to convert to a single strand polynucleotide chain. The amount of reassociation with similarly treated DNA from the another taxon which occur on mixing to is taken as a measure of similarity of nucleotide sequence. Then DLA polymorphism काम में ले रहे हैं. Utilization of DNA sequence data for phylogenetic relations analysis involve the identification of unique sequence which show certain variation in different organisms. These sequences which can be used to genetic marker and identification of target taxa and ultimately construction of the phylogenetic trees. इस तरह से आपके इसकी समरी आप देख सकते हैं कि किस तरह से हमने इस टॉपिक के हिसाब से हम देखेंगे कि साइटोलॉजी, पॉलीमॉर्फिज्म, सेल मॉर्फोलॉजी, कैरियोलॉजी, इन सब ब्रांचेज की सहायता से हम उसका एक सिस्टमेटिक पोजीशन दे सकते हैं इन कैरेक्टर्स का ध्यान रखते हुए. और इस तरह से नई ब्रांच टैक्सोनॉमी सिस्टमेटिक्स जो है वो वर्क कर रही है. तो ये जो सबको हम अलग अलग क्लास में लेंगे कि कैसे केमोटैक्सोनॉमी, एनाटॉमी इनका रोल है. और इस तरह से जो है आपका सिलेबस के जो पॉइंट्स हैं इनको हम अलग अलग क्लास में लेंगे कि ऐसे हमने इस तरह से साइटोलॉजी के आधार पे इसी जेनरा को क्लासीफाई करने में काम ले रहे हैं, फाइटोकेमिस्ट्री के आधार पे इसको ले रहे हैं. ये हम अलग अलग क्लासेज में इसको लेंगे. तो आप इनको देखिए और एक मिनट इस तरह से इसको करिए. और तो ये रहेगा आपका. और आगे जब हायर ये वाली रेफरेंसेज है, जब आगे क्लासेज में जाएंगे तो आप इन बुक्स को भी देख सकते हैं. हमने नई अप्रोच रखी है इसमें रेफरेंसेज की. ये हम इस तरह से बुक्स भी दिखा रहे हैं ताकि अगर आप कहीं ऑनलाइन भी ऑर्डर करें, कई बार बुक्स का आजकल ऑनलाइन का ट्रेंड है, तो आप ऐसे बुक्स को देखकर भी, और यहाँ लिस्टिंग है ही, तो उससे भी आप ऑर्डर कर सकते हैं क्योंकि ऑनलाइन डिलीवरी में घर आ रही है. तो आप कोशिश करें, घर पे रहें, सुरक्षित रहें और पढ़ें. नेक्स्ट क्लास में हम जो लेंगे नेक्स्ट टॉपिक इसी का और उसको डिटेल में लेंगे. थैंक यू.